Today I will talk about our recent ketamine study in which we try to understand how ketamine works in the human brain to exert its rapid antidepressant effects. In rodent models, injecting ketamine directly into lateral habenula attenuates the burst firing of these neurons associated with analogs of depressive behaviors in mice. Perhaps cognitively and computationally, some of the most notable works done on the function of lateral habenula was done by Matsumoto and Hikosaka using monkey neurophysiology and suggesting a role for habenula as a central hub for aversive learning. In humans, the seminal work in this area was done by Rebecca Lawson and colleagues using a Pavlovian learning task with aversive outcomes, in which they suggested that habenula encodes the expected values of aversive outcomes such as receiving electric pain. In our study, we used this Pavlovian learning task to probe human habenula and to understand how ketamine modulates this activity, but also with a view to translate the mouse model put forward by Yang et al. Study published in Nature. This is the outline of the study. We recruited 70 healthy volunteers without any psychiatric history, and we randomized these into intravenous ketamine versus placebo conditions in a 1-to-1 ratio. 24 hours later, participants completed the Pavlovian learning task inside a 7 Tesla MRI scanner, and approximately half an hour later we give them a behavioral assessment in the form of a preference test in which participants chose between all the shapes that they have learned about during the task, and there were 21 shapes in total. This is the timeline of the Pavlovian learning task. Essentially, this is observational learning without any behavioral input, and participants were asked to form associations between these abstract fractals and 4 different types of outcomes. In the task, we had winning and losing money as outcomes, as well as neutral shapes, which gave 100% of the time a null outcome, and finally we had shapes which gave electric pain probabilistically. Because the Pavlovian task does not have any behavioral input, the participants need to learn these associations through observation. We wanted to make sure that people pay adequate attention to the task, so 20% of the trials had these red flickers to which participants were asked to respond with a button press as quickly as possible. Because the Pavlovian task does not have any behavioral input, it's essentially like we are dragging the participants along an affective environment in which they observe these events, but they don't have any control over the outcomes. As a result, modeling participants' expectations of these outcomes becomes a little bit more tricky. In their seminal work, Rebecca Lawson and colleagues used the Riskola-Wagner model, and they reported that trying a number of different learning rate values does not seem to affect these results. But because we don't know how ketamine influences instrumental learning, we thought we can make a different assumption, and we used a beta distribution update rule, which suggests that humans will learn optimally from these abstract fractals and outcomes. And in this beta distribution update rule, on trial 1, participants would be indifferent with respect to the outcome probability, so it would be 0.5, 50%, but from there on they would be updating these expectations after each observation. For example, if they received electric pain or if they observed a no pain outcome. Technically, in Riskola-Wagner terms, the nearest learning rate equivalent of this process would be 0.124. But because the Pavlovian learning task does not have any behavioral input, we wanted to individually tailor these MRI regressors, and we relied on the preference test. So imagine on trial 1, participants would be completely indifferent with respect to the different shape types, but through learning they would come to understand that the wind shapes are the best and shock shapes are the worst. And we had a similar pattern of divergence emerge also in Rebecca Lawson's original findings. So in our final MRI regressor, we convolved this expected value signal with the perceived valence of the outcomes, such that we were able to linearly scale these expected values from the most appetitive to most aversive. The regions which were positively correlated with this regressor was the dorsal aspect of cerebellum and medial temporal gyrus. However, we were more interested in the regions which were negatively correlated with this regressor, because that's the kind of relationship which should probe human habenula. Here we had robust activities in the insula bilaterally, which is also a region implicated in pain processing and aversive learning, but also in a large cluster extending all the way down to habenula as seen here. Our pre-registered hypothesis concerned the habenular response to the shock outcomes during the expectation phase as well as the outcome delivery phase, and in order to extract this signal from the brain, we manually segmented the habenula from each participant's structural images, and we used those as ROIs. This subsequent analysis indicated that groups were mostly comparable. During the expectation phase, ketamine group shown in orange here, but during the outcome delivery phase, when the electric pain was actually delivered to participants' left hand, the ketamine group had significantly lower habenula activity in 2 out of 3 runs. Behaviorally, inside the scanner, the participants were mostly comparable in how quickly they were able to respond to those red flickers, where ketamine group had a slightly shorter response times. Outside the scanner, in the preference test, groups were also comparable in their behavior, although the ketamine group displayed in orange here had slightly higher preference for the shock shapes. And finally, during this preference test, the groups were mostly comparable for reaction times. Finally, we wanted to understand whether the neuroimaging findings relate to the behavioral findings, and what we discovered is that the average habenula activity during the expectation phase globally correlates with the average preference for the shock shapes, and this significant negative relationship indicates that higher the habenula activity, the lower the preference for the shock shapes would be. And this relationship holds true even if we control for the ketamine versus placebo group allocation, as well as subjective differences in pain intensity. And we saw this negative significant relationship even if we analyzed the individual runs separately, and as well as if we relied on other neuroimaging models. In summary, our study provides a successful translation of an influential preclinical model to human computational neuroscience. We show evidence to suggest that ketamine attenuates habenula response to peripheral delivery of electric pain during Pavlovian learning. Our study generally provides a clear role for habenula in aversive learning, such that if we reduce this activity with ketamine, it can inadvertently make negative memories less unpleasant. Therefore, participants select these more frequently subsequently in the preference test. This can also suggest that ketamine would have a feed-forward effect that can make patients with depression more resilient to negative events. I think finally our study provides information for preclinical models such that this habenula hippocampus pathway could be important for understanding ketamine effect on aversive learning and memory.